Well, we might as well start. It is a great pleasure for me to introduce Jonathan Chernoff, who came all the way from Philadelphia to give us a lecture. Jonathan Chernoff is a clinician scientist who does both clinical and basic research, which is very, very important these days for the medical school and for our health. He's actually a New Haven kid. He was born and grew up in New Haven and got his first taste for biochemistry and biology from Yale University, from Peter Langell, who was his mentor for the senior year at Yale. He graduated cum laude in biophysics and biochemistry in 1978. And then embarked on his career. 6 years later, he got MD, PhD from Mount Sinai School of Medicine in biochemistry. Later on, he got a residency in internal medicine from Pittsburgh Yale Health Center. Followed up oncology in Baltimore, Johns Hopkins, Baltimore, and then got some more of his basic education in basic research at Harvard University as a postdoctoral fellow. After that, he settled in Fox Chase Center and essentially climbed up the ranks from assistant to associate to full professor. He was very early recognized his talent in leadership and became the cancer program leader very early, follow up with what he is now, a senior vice president and scientific director, Ryman Chair in Oncology Research in Fox Chase. One of Jonathan's talents is to identify important topics in biology, in cancer biology, and in general. As a postdoctoral fellow in Harvard, he was with Raymond Erickson. He focused on phosphatases that are now really major field and major impact in cancer biology. He identified PTP, phosphatases, cloned PTP1B, and showed that the critical activation of circa through dephosphorylation of his inhibitory tyrosine 527 site that act as carcinogenesis. He identified several kinases, MST1, MST2, Pak1, Pak2, and delineated their function signaling in regard to cell proliferation and survival. He is the one that made the first knockout mouse model for Pak1 and Pak2 and used it to show that Pak1 is essential for KRAS-mediated carcinogenesis. Our pathway crossed about several years ago, about 1912, when we published that RAC1 has a recurrent mutation in melanoma, the p-21S mutation. And that immediately rang a bell with Jonathan because he worked on RAC1 for at least 10 years. And in a way, it justified RAC importance in carcinogenesis when you combine Jonathan's work and the recurrent mutation in melanoma. So he has about 164 publications through his young life and got several awards. I won't list all of them, but one of them is essentially 2013 third annual Drug Discovery Award from Pennsylvania Drug Discovery Institute, essentially on his work on p-21 activated kinases as targeted in cancer therapy. So today he is going to talk to us about p-21 activated kinases as target for breast cancer and maybe possibly melanoma. So here is Jonathan.
Thank you.
Thank you.
Well, it's an honor to be back here. You know, when I graduated in '78, I think I didn't speak at Yale maybe about once in 30 years. Martin Schwartz invited me a few years back to talk about some work that we were doing on heart function and cardiovascular function. But now this is my second talk here in a month. So I was here a month ago, and it's great. The challenge is that since there were a few people, at least in the audience, who were here for my talk in February, I don't want to bore you. I think that's rule number one for a speaker. You can't bore your audience. So you're not going to get that talk. It's got a different talk. I'm not going to repeat it. But there are a few things that you'll see. The first few slides may be the same, so please forgive me for that. If you hear a joke for the second time, you're allowed to laugh. It's okay, even if you've heard it once before. But for the most part, I'm going to be talking about something different. So last time, I talked mainly about KRAS signaling and a new method that's been developed to analyze the entire signaling set, all kinases at once in KRAS oncogenic signaling, which I think is a very interesting talk, but one I already gave. This one is going to be different. This is more about this protein kinase Pak1 that I've worked on for some time, and its role in breast cancer. Now, I did add in small letters melanoma because I think with Ruth as my inviter here, I would be wrong to leave out the melanoma data, but it's just going to be a small bit of that because it's quite new to us, and there's not that much to show, but I am going to show what we've got, just as a teaser, if you will. Now, before getting too much into the introduction, I did want to show this. I came across this picture recently, and it's fascinated me ever since I saw it. So this is a $10,000 bill from Singapore. Now, $10,000 in Singapore is worth a lot of money. It's not like $1 or $2. It's more like $8,000 US. So this is not a trivial bill. It's none that's not typically used for anything but perhaps the drug trade. But $10,000 Singapore, they honor their scientists on their currency. So you see we have a scientist who's looking through a microscope, and there's some structures around here. I'm not sure what this is, but some chip or something. But I was interested in this. I see these little peptides, and you can't help but look at them. And if there's some structural biologists here, you probably can interpret these pretty quickly. But what you notice, I focused in right on this because I knew this thing, ISLP. That's not unfamiliar to me. That is the key binding motif in this Pak1 kinase that I've worked on for years that allows it to associate with your protein RAC. And so why did it end up on the Singapore money? It's because I think Ed Mansour, who is one of the founders of the Pak field, works in Singapore, and somebody must have gotten this from him because I can't imagine why they picked this otherwise. But this is an extremely valuable protein. I want to leave you with that as a thought. Now, this work in some ways really started here. So Ruth mentioned that I had begun my work in signaling when I was still an undergraduate. So my first lab experience was with Peter Laniel. And at that time, Peter was working on these reovirus and its interferon response. And my project was to find the smallest piece of poly-IC, the smallest sort of artificial double-stranded RNA that would induce a protein kinase that in those days was just called double-stranded activated protein kinase, now called PKR. And interestingly enough, as we shrunk down these pieces of RNA down to about 22, below 22, we no longer saw this kinase. And of course, we didn't know it at the time. This is 1977. We're about 25 years before RNAi was discovered. But we were tickling the dragon, so to speak, but we just didn't know there was a dragon. So that was the minimum size of RNA that would induce this. And I mention this too because I see Dr. Weissman in the audience, and he probably doesn't remember me, but when I was an undergraduate working with Peter, it was your lab that supplied all of our ATP. So you literally had the hottest lab at Yale at the time. I think you were sequencing SV40, if I recall, and you were using p-32 gamma ATP to do it. And I don't know if all of Yale did this, but certainly Dr. Laniel, as I was an undergraduate, so I got sent on the mission to drive my car down to the medical school, pick up as much ATP as we could get from your refrigerator and take it back up Science Hill. Probably nowadays would be accused of being a terrorist or whatever, but that was how we did our assays with your ATP, so thank you for that. But that experience working with Peter got me interested in signaling, and I've never really gotten off of working on kinases and phosphatases. Anything that involves phosphate transfer, I like it. And it turns out that those pathways are so important for really all of biology, but certainly cancer biology. Cancer cells typically have aberrant signaling. In fact, I say they always have aberrant signaling, and almost always involving kinase pathways either as a primary target of mutation or affected by mutations in other genes. So when I arrived at phosphatase in the early 90s, I had made my name, if I had a name at all, in the phosphatase field as a postdoc. I had cloned this, one of the first phosphatases, as Ruth mentioned. But I was looking for some other things to do, and basically I needed another grant, and I wasn't going to get it working on phosphatases. And I got interested in this problem that I had first encountered in the Erickson lab where I did my postdoc. And I was remembering back to the work of a student who was in the lab, a guy named Craig Cruz, who I think some of you may know, maybe in the audience here. And Craig had, as I was leading the lab, was cloning, identifying MEK, the kinase that phosphorylated ERK. And that was a very important discovery for a lot of reasons. But for me, the main thing is it meant that these signaling pathways, which we had first encountered in yeast, like this one, which was the sterile mating cascade in budding yeast, which ended or terminated in a MAP kinase-like molecule, that since this pathway was also now, at least the bottom of it was seen in humans, it meant it was conserved. And believe it or not, back then in the early 90s, we weren't sure if signaling was conserved. It could be that every kind of organism had its own unique signaling, but when these pathways began to come out, both in fission yeast and then in man, it became clear that this was a very central and highly conserved pathway. And we started to get to work on some of the upstream elements. And the one that interested you the most was at this level, so sort of 3 levels up, or 2 levels up from the MAP kinases. And this one, fission yeast had a gene called BYR2, which was a protein kinase that phosphorylated BYR1 and spike 1 below that. Budding yeast had this pathway. But in man, it was a little different. So the RAF kinase had been discovered sort of as a RAS effector, but then shown to activate this pathway. And RAF didn't really look much like BYR2 or sterol 11. So that was a sort of a bothersome anomaly, if you will. And hope this is working okay. So we embarked on a screen to try to find kinases that looked more like sterol 11 or BYR2 that were not RAF. And it turned out, and this is a story I told last time, so I won't go into a lot of detail, but when we did our PCRs, which is the method we used, we got the homolog we were looking for, but someone else beat us to it. So Gary Johnson, who was in Denver at the time, just had published a paper on the cloning of MEKK, which was the very kinase we were looking for. And so, we lost that. But on the other hand, we got homologs to sterol 20, which was the budding yeast upstream, like MAP kinase to the 4th power, if you will. It was one step up from these sterol 11s. And we cloned them in fission yeast, we cloned them in man, and we actually cloned another family that was closely related called the MIS. And I was quite torn in thinking about what to talk about today, whether I should go into this. So these are extremely interesting kinases. So we called them MIS because they were mammalian sterol 20, they were mysterious, we didn't know what they did, so we gave them this name. And then about 5 years after we cloned them and did some biochemical characterization, they were rediscovered in flies as hippo. And of course, have become the subject of many, many, many papers. And we have some very interesting data on these MIS, but that will have to be for lecture number 3 if you ever can stand to see my face again here. So we won't talk about the MIS today, but I will focus in on these packs. Now, why are they called this anyway? We didn't actually give them this name. We had a different name for them. They got called this because at the same time we were cloning them, they were also being cloned by Ed Mansur, the guy in Singapore who put them on a $10,000 bill. So what Ed was doing was looking for effectors for small GTPases. So these proteins, CDC42 and RAC, are cousins to RAS, if you will. They're 21,000 Dalton small G proteins that had been discovered some time before and known to have activities in the cytoskeleton, but how they did it, totally unknown. And so he did a biochemical screen to find things that would bind to these. And he came across the very same genes that we had cloned by PCR. He got the naming rights because he had a function. So he could say these were p-21, the size of these proteins, activated kinase. And I'm careful to say that the definition because I once gave a whole lecture and at the end someone asked me about p-21 WAF and what its relationship was to this kinase. There is no relationship. This p-21 refers to these little GTPases. So Pak1, we cloned Pak2, we found the same in fission yeast and kind of have been working on these ever since. Now it turns out that there are 6 of them in total. there's a so-called group 1 and 2, or some people call them group A and B, 3 members of each. And structurally, the group 1s are pretty similar to one another and quite distinct from the second group. All the group Bs or group 2s are similar to one another, but somewhat different from the first group. And there's still a question in the field about, are these redundant or not? And our feeling is they're mostly not redundant. The more genetic experiments we do with knockouts and so on, the less redundancy we think we see. So I believe every one of these has individual functions. I think the same is true here. I'm not going to show you a tremendous amount of data about that, but I'm pretty sure that that's the case. Now, what do these things do? They do a lot of things, but as kinases, they phosphorylate things, so their substrates are one area of interest. They also have some scaffolding function, but in terms of sort of the classic characteristics of cancer cells, they help evade tumor suppressors. One of the ways they do that is by directly inactivating the Merlin tumor suppressor by phosphorylating it. They definitely have a role in movement, invasion, metastasis. Some of that may be through snail and limb kinase, which are also kinase targets of this thing, although this one I say is in doubt. This one is true. There's roles in angiogenesis, in metabolism, et cetera. And again, I'm not going to do an exhaustive review of all of these things, just to let you know that this kinase has many functions, as many central kinases do. Now, in my own thinking, I usually break it down into a fairly simplistic signaling diagram. Yes, there's many, many targets, but there's only a few that seem to matter in oncology. And these are the pathways that we think matter. So you have, on the one hand, a very well-established role in ERK signaling. So Pak directly phosphorylates RAF, it directly phosphorylates MEK. It does not do so in the activation loop, like you usually think of for these type of events, but it's sort of a permissive phosphorylation, if you will. Without that phosphorylation occurring, MEK doesn't couple well to RAF. Without Pak phosphorylating RAF, RAF doesn't couple well to RAS. So these are necessary but not sufficient phosphorylations. In most cell types, if you fail to do these phosphorylations, you fail to activate ERK completely. Now, that's not true in every single case, and actually last time I showed you one case where it wasn't true, but for the most part, that is true. So when you lose Pak, you lose this pathway. And so if you think about therapeutics, situations where this is an important pathway and you want to reduce it, it's another strategy that can be taken in addition to what people use with, say, MEK inhibitors or ERK inhibitors or RAF inhibitors. Whether it's superior or not, we can discuss, but it's another way you can get at that problem. The second pathway is, I'm going to actually show you data on, because it's fairly new, direct phosphorylation of beta-catenin and its role in cancer. And this phosphorylation, unlike the ones you usually think about from textbooks, is a stabilizing event. This is not the GSK3 beta destabilization of beta-catenin that is famous in the Wnt pathway. This is the opposite. A phosphorylation in a different site stabilizes catenin, more proliferation. And finally, and probably the least understood of all of these, there's some kind of connection between Pak and AKT-mTOR. We don't really understand this. We published a paper saying that Pak could phosphorylate PDK1, which is an activator of AKT. Another group said that wasn't true, that Pak acted as a scaffold and helped PDK sort of find AKT. We haven't really looked at this problem in a while, so I don't know how to comment, except to say if you take away Pak, you generally lose this pathway for one reason or another. And so that's a potentially important thing if you're trying to drug this. And it might change what sort of cancers you look at as well. So, typically, do you see this as on a list of cancer drivers? That people publish these papers, there's 100, 200 genes that people talk about. They don't always talk about Paks. It's not certainly on the top of the list. And the reason for that is, to date, there are very few cases of Pak point mutations that activate that are published. There was one paper a couple of years ago that Pak5, which is one of these second group of Paks, is mutated in a fair number of non-small cell lung cancer. And I put this asterisk here because that's one paper, and I'm just surprised that I don't see it more often in the databases out there. So I don't say that this is wrong. It's just, I don't know. There is a report, so there it is. But that shows you how rare these are. I look for these reports all the time. So in all my scouring literature, this is the only thing I can come up with. Now, just this morning, I found some more in a database locally here at Yale in melanoma, where there may well be other activating mutations and perhaps different Pak alleles. But we can talk about that next time when we analyze them. But what you normally see, or what you see most commonly, is the Pak1 gene and Pak4 gene are very commonly amplified in a bunch of different types of cancers. And I mean as much as 25% of breast cancer and ovarian, where this amplicon on chromosome 11, you can have 4 or 5, 10 copies of the Pak1 gene. And corresponding high levels of protein, corresponding high levels of activity simply by mass, I think. Not mutated, but there's a lot of it. Same is true for Pak4. And we've shown, and I talked about this a little bit in my last talk, is that on the other hand, if you get rid of Pak1, either by drugs, RNAi, or by gene disruption, you completely lose the ability of KRAS G12D to transform in an animal model and in vitro. And for these reasons, we think that this and some of its cousins might represent reasonable new therapeutic targets in several different types of cancers. Cancers driven by receptor tyrosine kinases because those work generally through RAS. Ones driven by KRAS, at least this type of KRAS. And if there were any tumors driven by RAC, which at the time we started this project we didn't know about, they would be prime candidates for Pak inhibitors. Now, are there such tumors? I think there are, but we didn't know about them until very recently. And so what I'm going to talk about then in the next little while is a very brief vignette about melanoma, because that, had we known about the RAC mutation 10 years ago, we would have worked on this first, because it's the most obvious case for where you would use an inhibitor for this kinase, since it's directly activated by RAC. But I'm going to spend the bulk of my time about breast cancer, which was, after all, the advertised title of the talk about HER2 positive breast cancers, the role of Pak there, and also in these cases where the gene itself is amplified, and what role, if any, Pak has as a driver in that setting. Now this is a lattice sequence. Okay, so this is directly from work here at Yale. I think I have your numbers right because I quoted from your paper that about 5% or so of melanoma patients have RAC1 activated mutations, generally associated with UV damage. And at least from my simple-minded view of signaling, there's really two targets to think about in that disease. If it's driven by this, if this is an important mutation, Pak is, one of the most famous kinase, protein kinase effectors, but there's a lipid kinase that's specifically associated with RAC, PI3 kinase beta, which would, I think, have equal weight as a potential target. We ourselves have not done much with this because we don't have the kind of genetic models that others have, but this is, if I'm thinking about melanoma driven by this, I'm thinking about these targets, just to put it that way. And this is just a spectrum of mutations. For whatever reason, but perhaps people here know better than I, this one mutation by far dominates p-21S, creates a fast cycling version of RAC, and there's also could be p-29L between those two. That's the majority of cases. And then there's a scattering of other mutations. I'm not sure all of these have been analyzed in great detail. So, well, either. What would happen if you started monkeying with these pathways in melanoma? And this, again, I only have just a few slides on this because it wasn't the planned topic, but we acquired from Ruth and from Maynard Herlin at Wistar some cell lines that had this RAC mutation or did not. Now, many of the cell lines are complex. They have more than one mutation. They have RAC plus something else oftentimes. And that's the case here, where some of the cell lines have RAC, but they also have NRAS or other mutations. And if you ask them to grow on a plate, they grow. They all grow, pretty nicely. If you treat them with a MEK inhibitor, at least in the short run, they all get inhibited. Whether BRAF mutant, RAC mutant, NRAS mutant, they all slow down, which I think we had expected. But the interesting part comes here. So 3 of our cell lines had this RAC mutation. 2 were just BRAF and not RAC. And we gave them a Pak inhibitor. I'll talk about it a little bit later. It's a pretty good compound. And it profoundly affected proliferation of these RAC mutants. Did not really touch the BRAF alone mutants. And as you might guess, we did the reciprocal with a RAF inhibitor and got reciprocal results. So this drug did quite well in the short run against BRAF mutant cells. Did really nothing against NRAS alone or NRAS in combination with the RAC mutation. So again, this is just an in vitro experiment with a few cell lines, but it's suggesting that the RAC mutants have a different sort of signaling profile and may respond to different types of inhibitors. And the signaling, I don't want to get too deep into this, but if you look at the lines that had this RAC1 mutation, they are very strong PAC activators. That's no surprise. If you have active RAC, you're going to have active PAC unless things are really crazy. And everything else is on. AKT and ERK are on in these cells, as you might expect they would be. But when you start treating with inhibitors, interesting things happen. So remember, the BRAF drug did lousy against RAC mutants. And you see it didn't inhibit ERK at all. So it did very well in the two BRAF lines. But whenever this RAC mutation was present, or I suppose you could blame it on NRAS as well, you can't really tell here, but this one is on. Of course, it didn't inhibit PAC, because why would it? BRAF inhibitors don't inhibit PAC. And conversely, the MEK inhibitor did well no matter what your mutation was in terms of ERK. Didn't touch PAC in two of the lines. And the PAC inhibitor, on the other hand, did quite well in inhibiting ERK in RAC mutant lines. So that's really all the signaling I'm going to show you here, because we have some more, but it's just not ready for prime time. Even this isn't really ready for prime time, but it's just a teaser that we think, again, these signaling are wired differently if RAC mutation is present, rendering them potentially sensitive to PAC drugs, if such drugs could ever be developed clinically. And just finally on this melanoma story, we have been scratching the surface at doing some mouse models and other animal models. And although we're partway into making a mouse model, we did a quick fish model first to see if we could verify anything in an in vivo setting. So we used sort of typical technology in zebrafish where we could make a transgenic RAC1 mutant attached to MID-F, inject embryos. You know they're transgenic because they rescue melanocytes. Normally they wouldn't have them in this background. And indeed, we get melanoma in that setting as long as p53 is mutant. And the interesting thing is what happens if you add a PAC inhibitor to this. I think, I'm not going to show this unless there was some effect, right? They did well. So again, this is not a definitive experiment. We didn't analyze any signaling in this fish at all. It's not an N of 1, but it's not an N of 100 either. It looks like you can block RAC signaling in vivo in this fish model. They said, we're going to do this in the mouse soon enough, but it just takes longer. So that's the melanoma stuff. I'd be very happy at the end if people want to talk to me about it. There's not more here today because of just where we're at in that project. All right, so now I'm going to turn to the main topic, which is breast cancer. Now, this diagram was generated in this famous paper from Curtis et al. In Nature a couple of years ago, where they were looking using genomic profiling to try to divide breast cancer into genotypes that had survival information. And so they called these things integration clusters, and they had different definitions on what made their 10 clusters. And the two that I'm going to focus on, because I think PAC has a role in both of them, are the two worst actors of the bunch. So in class 2, that's essentially defined by that amplicon that I referred to earlier, this 11q13 amplicon where the Pak1 gene sits. That is essentially what makes this cluster. So those are a really bad prognosis breast cancer compared to others. And then in class 5, which you would otherwise know as HER2 positive breast cancer. That's pretty much the definition of in class 5. So those are two bad actors. And I'm going to show you a model first with the in class 5 or HER2s, and then with the in class 2s to show what role, if any, PAC inhibitors might play in those diseases. All right, so here's your typical signaling diagram, pretty much similar, whatever receptor tyrosine kinase you like. In this case, we're talking about the HER2/3 heterodimer and the usual signaling apparatus below. I placed PAC here, showing its role in activating MEK and also RAF and AKT. I did not show beta-catenin on this because I think at the time we didn't know about that. So our initial thinking was if HER2 tumors were at all responsive to PAC inhibitors, and we weren't sure they were going to be, it would be due to inhibition either on this side or on the AKT side. And so we decided to look at that. And we took advantage of a system that developed by Joan Brugge, building on earlier work from Minabissel, which is a 3D culturing system that some of you may be familiar with. This is using the MCF10A cell line, but you can do it with other lines as well, where you take these relatively normal breast epithelial cells, put them in matrigel, and they grow in this very nice sphere. And they hollow out the lumen. You can see that better in this diagram. After 14 days or so, or 10 days, they stop growing. The lumen gets hollowed out, and they actually start producing casein. So they're behaving like a mini gland. And they're very stable at this point. You can leave them in matrigel for a long, long time. It's basically an organoid culture. And what Joan had done with her postdoc, Senthil Muthuswamy, had put in an artificial version of the EGF, of the HER2 receptor. So they wanted a version of this receptor that they could control with a ligand. So they made one of these constructs where you could take this compound, AP1510, cross-link the HER2 and cause an artificial heterodimer, or a homodimer in this case, and then drive this nice normal-looking acinus into this awful-looking, sort of cancerous-looking lesion, which has a filled lumen and a very irregular surface. So that's a model. I didn't develop it, but I did do my only sabbatical of my life so far in 2003. I actually went to the Sentinel's lab at Cold Spring Harbor for about 6 months and did a lot of this work myself to learn the system. And some of this data may even be mine. I don't remember anymore. But so one of the things we did right off the bat was just ask, are Pak1 and 2, which are the most commonly expressed alleles of these Paks, are they involved in any way in the growth of these structures, and do they have anything to do with HER2 signaling? So we set up some a tetracycline controlled hairpins, which work very nicely to inhibit either Pak1 or 2, and asked would they affect growth rates at all. And it turned out that when you turn on HER2, which is the top line here, they grow faster, and that's, of course they grow faster because you now have an oncogene switched on, forcing these cells to grow faster. So this is a control. If we knocked out Pak1, or knocked down Pak1, now HER2 really didn't have any special effect. Whether it was on or off, the cells grew at some modest rate. And if you knocked down Pak2, it had no effect whatever. So now the cells grew very well when HER2 was on, less well when it was off. And this this is actually apoptosis, where we got the opposite type result, where Pak1 had an effect, Pak2 did not. So that was sort of interesting. As I said, maybe Paks have a role here, but only Pak1. Now here's what some of the architecture looks like, because these things are really best for looking at architecture. That's why we grow them in 3D and stain them. So this is a normal MCF10 cell growing in matrix gel. It forms these small acini. You stain them for actin. You can see the actin structure. It looks nice. Caspase, you see these red dots. That's caspase, mostly in the developing lumen. So they're carving out a hollow shell in the middle of these balls of cells. And if you look at Chi67, it's generally off when the acini reach maturity, they don't proliferate anymore. So you don't see any more staining. If you turn on ERB2 by using this artificial ligand, now the acini look completely messed up. They're no longer single, they're sort of multilamellar, very irregular. They grow in these clumps. The actin just reflects what you're seeing here. If you look for caspase cleavage, there is very little because it's unable to carve out a nice lumen anymore. So you have these are filled spheres rather than hollow. And look at Chi67, it's lit up like a Christmas tree. Like they don't stop growing even though they were supposed to. So it's clearly this oncogene does bad things to these cells. Again, no big surprise. Now what happens when you start monkeying with Pak activity? So we have these hairpins that we can turn on and off. And just to run through, the top is a control. Normal, we switch on HER2, they look awful. You switch on HER2, you lose your nice lumen, you lose your caspase. You switch on HER2, they grow like crazy, even though they're supposed to be like this, but now they're like this. So that's what I showed you before. If you now put in a Pak hairpin, what happens? Well, it blocks everything. So we switch on HER2, but in the presence of this hairpin, the acini look just fine. They're carving out lumens just fine. They're stopping growing just fine. So they're essentially like they don't have HER2 on at all, even though they do. On the other hand, if you take out Pak2, none of those good things happen. You don't block HER2's effect on morphology. You don't block the effect on caspase, and you don't block the effect on proliferation. So it's as if this is irrelevant, doesn't matter at all. This one matters. And again, that's consistent with some of the growth I showed you in the previous slide, but now in a little more detail. Now what about signaling? And this is really where our nice sort of comfortable story went down the tube. So here is a signaling version of what I showed you on the growth slides a minute ago. And just to run through some of this, we're looking at the most popular signaling targets, ERK and AKT, et cetera. And as we expect, when we put in this Pak1 hairpin, we get rid of MEK and ERK, and AKT for that matter. And that's what we expected because we think Pak1's involved in all those things. But much to our surprise, when we got rid of Pak2, we had equally good effects on all of the signaling pathways. Now, that basically cannot be, because I just got through the trouble of showing you all these slides saying that knocking out Pak2 had zero effect on any growth parameter, and yet these very important signaling pathways are completely suppressed, or as well suppressed by Pak2 loss as Pak1. So that is weird. And here's just knockdown of Pak1, but not Pak2 impedes transformation in this in vitro setting. But you lose either one, you get the same signaling effect. And this led to a lot of puzzlement. And the only thing we could come up with is that we had just been focusing so much on ERK and AKT that there must be some other difference, because those aren't different. That there must be other targets that we're just not thinking about that are different between Pak1 and 2, and we needed to look at those, or else there was no explaining anything. And so we turned, actually I'm going to get back to that in a second. We also did an in vivo version of this, where we took a mouse model. This is a rat version of HER2 called Neu, which is also oncogenic, bred it to our Pak1 knockouts. We didn't do this to Pak2 at the time, we didn't have them, but we just asked, would things improve if you lost the Pak1 gene in terms of cancer initiation or progression or survival? And it turns out that our Pak1 knockouts survive better, although not indefinitely, than the controls. So they gain about 5.5 months of life, which is a lot for a mouse that only lives 2.5 years or so. So there was a benefit to losing Pak1. My prediction would be there'd be no benefit to losing Pak2, but we didn't actually do that experiment. And actually the signaling was exactly the same as before. We lost Pak1, we lost Pak, of course, but we lost ERK and AKT. AKT is actually pretty bad either way, but we lost those pathways both by IHC and by Western. So we were forced to finally turn away from our friendly, familiar pathways into looking more broadly. And at this time, this is before we had gotten seriously into this kinomics that I talked about last month, and we used a different method. So there are companies out there that sell phosphoantibody arrays. This one was called either Blue Moon or Full Moon or some kind of moon. I don't remember exactly, but I do remember the slides were about $1,000 a piece, which I didn't appreciate. And I remember when I gave this to the postdoc to do, I said, you get 2 slides and you better do this in 2 because we're not doing it anymore. And they actually made a lot of use of those 2 slides. I think it was probably money well invested. So this particular slide had a bunch of proteins on it. Not all that I would have wanted, but it's what they have. Now people do this RPPA method that you can send down to MD Anderson. It's essentially similar to that. And so what we saw, what you're looking at here is cells derived from this in vivo model that have either lost Pak1 or not lost Pak1. So this is derived from in vivo setting, not from a cell line. And when you look at this line in the middle, this would be no change whether Pak is present or absent. If you're on the left, it means you're less phosphorylated when Pak1 is gone. If you're on the right, and there's not too many on the right, you're more phosphorylated. And so the ones I've written in red were targets that we knew about. Like RAF is much less phosphorylated when Pak is gone. And we predicted that because we know that RAF is actually a target of Pak. Bad, we knew that was a target too. I didn't talk about this, but it's an apoptosis. It's important. AKT was less phosphorylated. Again, all predicted. A bunch of other ones that we didn't comment on. But the one that struck me was the one on the very top of the list, beta-catenin. That was not on our list of things to think about at the time, and yet it was massively under-phosphorylated when Pak was lost. So we started thinking about that. And this is your canonical pathway that there's in all the textbooks. You have beta-catenin, it gets destabilized first by CK1 and then GSK3 beta. It then becomes subject to ubiquitination, disappears, goes away, and no signaling. But there's also a smaller literature on other phosphorylations of beta-catenin, including the 675 site, which is just the opposite. This one prevents this from happening, leads to increased beta-catenin activity and more target. And it turns out that Pak, this is a perfect Pak consensus site, and it is a Pak site. It phosphorylates it in vitro, it does it, we think, in vivo, and we think it explains the role of Pak in the catenin pathway. So, our addition would be this, that ERB2 or HER2 somehow talks to Pak, and it's pretty easy to figure out how that happens, and then Pak directly phosphorylates catenin. But is it true? Well, interestingly enough, for two famous proteins like HER2 and Wnt, two famous pathways, there's not that many papers that talk about them at the same time. Usually when you Google something or do a medline, for whatever terms you put in, something pops up. This was one of the few cases where very little popped up, considering the fame of the two search terms. But, so we did some basic experiments that probably should have been done earlier. What happens to beta-catenin anyway when you stimulate HER2? Well, I guess, it goes to the nucleus and activates. But that was not well published, let's say. But we saw it in our system that when we stimulated with a Wnt ligand, we could drive catenin into the nucleus and on. And similarly, when we cross-linked this 3D system we had, it wasn't quite as potent, but we did get catenin in the nucleus. So that's just kind of a control. What happens, no, I'm going to skip this. What happens when we start monkeying with Pak levels? So what happens, here's a normal acinus, and this is a 3D system. When we turn on this oncogene, of course, the acinus looks messed up. We actually see quite a bit of catenin staining here. If we knock out Pak1, we don't see any catenin at all, neither at the membrane nor in the nucleus. If you knock out Pak2, nothing happens. So this would, and this is actually shown here too. So here's a Western blot. Knock out Pak1, we knock out the gene, or knock down the gene, lose a lot of catenin. We also lose phosphocatenin, not shown on this blot. Knock down Pak2 very nicely, no effect on catenin. So this is different than the ERK story. I showed you before that whether you lose Pak1 or 2, ERK and AKT both go down. This case is different. Only Pak1 is affecting catenin phosphorylation, not Pak2, whatever. And we bought, since we had such success with our first slide, I said, let's buy more slides. So there's a slide that you can buy that has Wnt targets on it. And you can measure a whole bunch of Wnt targets at once. Every time I look at this, I think another 1,000 down the drain. But it wasn't down the drain, because what we saw is almost all our Wnt targets were down. They were down-regulated when Pak1 was knocked down. And if you looked at your favorite Wnt target, instead of all of them at once, you can look at things like MYC. That wasn't as impressive, but when you lost Pak1, MYC expression is down, which is a catenin or Wnt pathway target. Cyclin D is also down quite a bit. And that was not true at all with Pak2. Now, all of this is done with shRNA, so that is not a perfect way to study. So we decided we'll do some drug studies too. And at this time, the best inhibitor for Pak around was this Pfizer compound. Now there's much better ones. But this Pfizer compound, in fact, if you use it, you lose Wnt signaling. And this doesn't prove it's going through Pak, but it does show that your cyclin D and MYC levels go down, and your catenin certainly is going down when you use what is thought to be a Pak inhibitor, although not a very clean one. And it's not just this one cell line. I think we were sensitive to the fact that almost all our experiments were either in vivo, which is good, or in this 2A, this MCF10A cell line, which is just a single cell line. So we did some other patient-derived cell lines, which are HER2 positive, using the dominant negative expression of Pak1, which is sort of an old-fashioned technique, or using this drug. And in each case, we said almost total loss of beta-catenin, whichever approach you want to take. And speaking of the compounds, this is sort of what's out there today. So since those studies were done, there's a company in San Diego called Afraxis that was interested in Fragile X and other syndromes like it, where Paks are thought to play a role. And they had developed a compound called Frax 597. This was a pretty bad compound. This is tyrosine kinases up here. If you see a red sphere, it means it's highly inhibitory towards them. So when I looked at this, I said, what you guys have is a tyrosine kinase inhibitor that has off-targets on Pak. They were calling it a Pak inhibitor with off-targets on tyrosine kinases, but whatever. We used this. They improved it. And then Genentech bought the whole program from them and improved it some more. They have a compound now, it's a little messed up here, but we call it Superfrax. They call it G5555. It has a very nice profile. It inhibits Pak 1, 2, 3, not 4, 5, 6 at all. Has a couple of other off-targets and basically very little else. So it's a very nice compound, and it's the best one we think that there is out there at the moment. Now, Pfizer has not been silent either, although they're not sure how active they are. This was their original compound, which had a lot of off-targets. There's the Paks right there. This one inhibits all Paks, 1, 2, 3, 4, 5, and 6. A lot of off-targets. They've improved it somewhat since then. We have these compounds, but we haven't done too much with them. But that's sort of a major companies where they're at with this. Novartis has a program as well. But just getting back to the use of these and their help in our analyzing signaling, we now wanted to know, since we had ID'd Wnt signaling as somehow involved with Pak, that Pak was controlling how much catenin you had, that suggested to us that if you block the Wnt signaling, if you block catenin itself, that might have beneficial effects either alone or in combination with Pak inhibitors. So we're going to test that. So here's the same cell system that we were using before. Nice acini, messed up acini. We turn on HER2. If you add catenin into this system, the cells are not liking it. They actually look kind of transformed even before we add the ligand for HER2. And that's, again, not too surprising. Wnt or catenin is generally a positive factor in cell growth. If you add a bunch of it into cells, you might expect them to take on sort of a transformed phenotype, and they do. If you put in a special form of catenin, which is truncated and highly active, it's even worse in this assay, so to speak. It causes the acini to look even worse than before. But the key thing is if you put in a small molecule inhibitor, which do exist, at least for researchers, it completely blocks, first of all, it has no effect on its own, and it blocks the effect of HER2, just like loss of Pak1 might do. And we think for the same reason. Now, if you ask formally about synergies and start treating cell lines with Pak inhibitors plus or minus these catenin inhibitors, what you get is true synergy, at least by the mathematics, with CIs below 0.5 in most cases. And if you ask, will you sensitize resistant cells that have, don't normally respond to HER2 compounds? The answer to that is yes. So here is the combination, well, either one drug alone, the Pfizer drug, which is a Pak inhibitor, the beta-catenin inhibitor, and the two together. So in this cell line system, they seem to work together better than either would work apart. And if you treat this is a xenograft, I believe. Yeah, xenograft with one compound or two. Actually, the catenin inhibitor alone in this setting was quite effective. Adding the two of them was marginally more. I think if we could have done this experiment again, we would have lowered the dose of the catenin inhibitor. But they were definitely suppressing growth of this tumor quite a lot and suppressing signaling in the way that you might expect them to. And this may be the last slide of this series. That addition of the two compounds together gave you more than just a little bit of apoptosis. It looks like you're getting sort of necrosis in the tumor tissue now when you add these two inhibitors together. So I don't want to make more of that than it is. It is just an experiment to attempt to find rational combinations. But what I've just told you in this whole section is that Pak1, although it affects, and it's definitely involved in activation of ERK and AKT in HER2 breast cancer cells and in an animal model, we don't think that's really critical, which is surprising. I would have predicted differently before. What we do think, and that's why I made a fat arrow here, is that Pak1 is very important for catenin activity in that setting. And if this is broken up in any way by either inhibiting Pak or inhibiting catenin itself, you really have a powerful effect on the growth of these cells. That's just summarized here. Now, we do have some recent information that in these inhibitors, we may not want to be messing around too much with one of the isoforms, Pak2. This seems to be a problem child in that adults need Pak2 to survive, we think, or at least mice do. So future inhibitor development, we're trying to push more for isoform specific because this one could be a problem. And again, that's something I can't talk about too much now, but there's some interesting data that we're accumulating on that. All right, now the last couple of minutes, okay, yes, just a couple of slides on this. I said I was going to talk a little bit about this one other type of breast cancer, that is those that have this 11q13 amplicon, which are almost equally bad to the HER2s. Now, if you look at this amplicon carefully, the Pak1 gene is here, and it used to be thought that cyclin D, which is also on this chromosome, was the driver in this amplicon. And as people have gotten better and better about mapping and just measuring levels of gene amplification, it's pretty clear that there's possibly multiple drivers in this region. And so the question has arisen, is cyclin D important, or any of these other genes in red important? Is Pak or GAB2 or what, MZ? There's a lot of genes that people, if it's your favorite, it becomes the one that you think is driving, but nobody really knows. So we wanted to have an organized approach to this. Was there some way that we could just look at this problem in total? This is my fanciful diagram of just taking all the genes in red. Can I put them in a pathway? This is sort of my own version of a Go analysis. Yes, I can put them in a pathway. That doesn't mean it's a real pathway, but I can draw it. And certainly, GAB2 talks to receptors, talks to Pak. RSF1 has some roles here in ERK activation. Again, I think this is almost worthless. It's just, playing games with the genes that are on that chromosome. So we wanted to get away from sort of a priori things like this to just analyzing the pathway in a non-biased way. And that had been done by some people before. This is an ovarian cancer, which is not breast cancer, but people had looked at the same amplicon in ovarian. And this guy, who is at Hopkins, was using RNAi to try to see which of his favorite genes might be important. And he came across one, I think it's RSF1, that he felt was the cells were particularly sensitive to loss of that. And that's ovarian cancer. He did do Pak1 in here. It didn't seem to have much effect. But our own work on Pak1, which is shown here in ovarian, showed loss of Pak1 actually had a very profound effect on cell growth. I'm actually going to skip through this in the interest of time just to get to this little project we decided to do. So all of that's to say there's a question about who's driving what in this amplicon. We have our favorite, other people have their favorites. And so we decided to just take a non-biased look at this. And so we decided we'll just use, CRISPRs around. You can knock out whatever gene you like. Let's just knock them all out one by one. So we thought about making CRISPRs for every gene in that amplicon. And there's about 300 genes. Not an impossible number. And we thought, well, we'll do a traditional dropout screen. If you did this and something was really important, like cyclin D, for example, or Pak, that cells wouldn't grow well if you knocked out that gene. So therefore, at the end of a couple of weeks of growth, they'd be underrepresented. And you can count that with a sequencer. People do this sort of thing. So here's the screen. You take some breast cancer cells, in this case with the amplicon, you put in your library, select for everybody who's green, wait a few weeks, PCR up whoever's left, and just count, knowing that they all started off equally in the same amount. And so, why would this not work? Well, there's a lot of copies by definition of each allele. So CRISPR is usually people, they're getting 2 alleles, but can you do more than 2? Maybe RNAi would be a better approach. This is from a paper from George Church that says you can do this. So he had looked at these pig cells that have 62 copies of some porcine retrovirus that he wanted to get rid of for transplant purposes in the future. So he put in a CRISPR, and all 62 copies got cut. We're not asking for 62. We're only asking for 5 or 6 copies. So we think it can be done technically. Now, we may not be as good as George Church at CRISPR, but if he can do it, then others can do it. And then is it quantitative? Because for this to work at all, you have to it has to work out, sort of the math has to work out. So this is just, this isn't what I learned at Yale, but what I learned at Edgewood School. If you do arithmetic and you have a 10% loss of growth every cycle and you go through enough cycles, after 12 generations, only 25% or so will still be there at the end of that time. So that's sort of the theory behind doing these dropout screens. And so, well, that's just how you do it. It turns out we can very nicely cut all the Pak1 alleles in this cell. That's not a problem. We test everything in vitro first to make sure our CRISPRs work. We cut DNA like it was a restriction enzyme, so we test them to make sure they work. This one didn't work so well. This one worked really well. And so here's the result. I mean, this is very early because we just started this, but for our first cell line, and it's very interesting. So we used SUM52 cells, which are just one of the cells out there that have this amplicon. MCF7 is our control, does not have the amplicon. Went across the entire array, and it turns out who scores top? Cyclin D, just like people had thought. And Pak1 is pretty close for number 2. There are a few other genes I won't talk about here that seem to also score, but here's sort of a map of where these things are. We were very impressed with sort of the power of this screen because, of course, we have to do it in a lot of other cell lines to make sure that this comes out the same. But at least in our first test, we're getting things that we expect for a change. And that means that if you can do it on this amplicon, you can do it on any amplicon. I mean, we're interested in this one, but there's a lot of amplicons with unknown drivers. And I don't see any reason why one couldn't do the same sort of thing to analyze who's important for any given polyploid cell. And in particular, one that we studied, we did confirm that these are likely drivers. There may be a few others there too, but these seem to be the main 2, which are the 2 that people have focused on for the most part. So with that, I'm going to stop. There's a lot more I could say, but with the hour's up. The work in my lab was done mostly by these people. I won't identify each person with what they did, but by crew. James Duncan, I featured him more last talk. He's a guy who does a lot of our kinomic work on analyzing total kinomes. I didn't say much about it. Pfizer, Afraxis, Genentech supplied us with tons of different compounds. We couldn't have done it without them. And new collaboration, we didn't talk too much today, but Ruth, certainly, we hope to work with you. And Maynard in Philadelphia, it's close by, we have gotten some cell lines from him. So with that, I'm going to stop. I'll take any questions. Thank you.